Welcome to In-Silico Trials Real Impact, the In-Silico UK podcast that's revolutionizing the way we think about medical innovation. I'm Professor Alejandro Frangi from the University of Manchester, and I'll be your guide through this fascinating world of in-silico technologies. In each episode, we'll dive into cutting-edge research and groundbreaking news that are shaping the future of medical product development and regulation. From computational modeling to simulated clinical trials, we are exploring how digital approaches are transforming healthcare, making it safer, more efficient, more equitable, and incredibly precise. So, whether you're a researcher, regulator, a member of the public, or a patient representative, or simply curious about the future of medicine, tune in as we unravel the complex world of in-silico methods and their real-world impact. Get ready to discover how computer-simulated trials are leading to very real breakthroughs in patient care. In-Silico Trials Real Impact, where algorithms and data are saving lives. Let's begin.
All right, welcome back to the deep dive. Today we're diving into something pretty crucial in the medical device world, and it's getting more and more important.
Yeah, definitely.
We're talking about in silico clinical trials, ICSTs for short.
Catchy, right.
Yeah, exactly. And it's all about using computer simulations, powerful ones, to really assess these new devices coming out.
And it makes sense too.
It does,
Yeah.
especially when you think about how expensive traditional trials are getting.
Oh, tell me about it.
And the ethics of it all, you know. You're talking about class 2 and class 3 devices here. That's serious stuff.
No doubt. You got vulnerable populations, kids, rare diseases. It's tough to get enough people for those traditional trials sometimes.
Right. So that's where in silico trials come in. Potentially, anyway. They might be able to complement or even replace real patient data.
That's the big hope, and it could really shake things up.
Huge implications for sure. But for our listeners out there, especially those navigating the regulatory side of things, they got to understand what makes these virtual trials tick.
Absolutely. And that's where VVUQ comes in. Verification, validation, and uncertainty quantification.
VVUQ, right? Sounds a bit jargony, but I think it's the heart of making these computer trials credible, right?
It really is.
So
Yep.
let's break it down. What's verification all about when we're talking about these complex models?
Well, think of verification as making sure the model is doing its math correctly, solving those equations the way it's supposed to.
So we're checking if the code is actually reflecting the math accurately.
Yep. And if we're getting the right numbers based on how the model's designed, it's all about internal correctness.
Got it. So it's kind of like double checking your work on a giant, super complicated problem, making sure there are no mistakes in the process.
Exactly.
All right. And then there's validation. This is where the virtual world meets the real world, right?
That's the one. It's all about asking, does our model match up with what actually happens? And to answer that, we compare the model's predictions to real-world data.
What kind of data are we talking about?
Everything from physical experiments to preclinical studies, even human patient data.
So the gold standard.
You got it.
All right. So verification is about the model getting the math right, and validation is about it getting reality right. That's a good distinction.
Yeah, that's important.
And finally, uncertainty quantification. I'm guessing that's about understanding how much we can trust those model outputs.
That's a great way to put it. With UQ, we're looking at all the things that could make our model uncertain.
Like what?
The inputs we feed into it, assumptions we make, even uncertainty in the data we use for validation. We got to figure out how all those uncertainties affect our final results.
So basically, knowing how much faith we can put in those predictions.
Exactly.
Now, for our listeners, especially those in regulatory affairs, today's deep dive is all about understanding why this VVUQ thing is so important for getting in silico trials accepted by regulatory bodies. It's about showing them these models are credible, right?
Couldn't have said it better myself.
And I have a feeling we're going to have some aha moments along the way about how these concepts actually impact those crucial regulatory decisions.
I think so too. Let's get into it.
All right, let's start with why in silico trials are becoming so important.
Well, one big reason is money. Traditional clinical trials, they cost a fortune.
No kidding. And the ethical issues are huge too.
Definitely.
Sometimes you're trying to test devices on kids or for rare diseases. Finding enough patients for a traditional trial can be almost impossible.
Exactly. That's where in silico trials have the potential to step in, right? They could help us overcome some of these big challenges.
Right. Plus, simulations could give us more statistical power even if we have smaller, real patient groups.
That's right. And don't forget about patient safety. We can reduce the number of people exposed to a new device if we're using simulations.
So to sum up, it's about lower costs, fewer ethical dilemmas, potentially better data, and safer for patients.
You got it.
Sounds like a win-win, at least on the surface.
Yeah.
Now, what about the regulatory side of things? Are they on board with all this?
Well, both the FDA here in the US and the European authorities are definitely paying attention.
Good to hear.
The FDA's even issued guidance on reporting simulation results, and they're working on industry standards for model credibility.
That's a good sign, right?
Oh, yeah, they're taking it seriously. And over in Europe, the Avicenna Alliance has a formal definition for in-silico clinical trials now. They see it as using personalized computer simulations in the development and evaluation of medical devices.
So it's not just a futuristic idea anymore.
Nope, it's becoming reality.
All right, but here's the thing. If we're going to use these simulations to make decisions about medical devices, about people's health, those models better be darn reliable.
Absolutely. Model credibility is everything.
If the FDA and others are going to use this data, they have to be confident that the model is giving them an accurate picture of the real world.
Exactly. It's the same principle as traditional trials, right? We need strong evidence to ensure patient safety and make sure the device actually works.
Makes sense. So VVUQ, that's how we build trust in these models.
That's the key.
Now, I read that the level of rigor in VVUQ, it's not a one-size-fits-all thing.
Right. It should match the level of risk involved.
So if a model's predictions are going to be used for something really important, like approving a life-saving device, we need to be extra careful.
Right, exactly. The higher the stakes, the more rigorous our VVUQ process needs to be. And there are standards for that, like the ASME V&V40, that specifically link model credibility to the level of risk.
Makes sense. We wouldn't put the same effort into validating a simple model as we would a complex one that's guiding decisions about a heart implant.
Right.
All right, let's dig a little deeper into VVUQ, starting with verification.
Sure.
We talked about it being about the mathematical correctness of the model. But how do we actually do that? What are the steps?
Well, there are two main parts. First is code verification. That's making sure there are no errors in the code itself, that it's implementing the model's equations properly.
So no bugs, no typos in the code.
Exactly. And one way we do that is by comparing the model's output for simple cases to analytical solutions, which are like exact mathematical answers we can calculate separately.
So it's like checking the code against a known answer to see if it's working right.
That's the idea. Then there's solution verification, which is about the accuracy of the numbers the model spits out.
So making sure the model is actually giving us good numbers.
Right. We check things like resolution, making sure the model has enough detail, and convergence, which means making sure the solution stabilizes as we refine the model.
Okay, so we're making sure the model isn't changing its mind all the time as we give it more information.
Exactly.
Now, the source mentioned that verification often only needs to be done once, as long as the numerical methods stay the same. Why is that?
Well, if you're using the same math and the same software, and you've already checked that it's all working properly, you don't need to start from scratch every time you use the model.
Unless you change something significant, right?
Exactly.
That can save a lot of time and effort.
For sure.
All right, let's move on to validation, where the model goes head-to-head with reality. What kind of real-world data do we use for this in the medical device world?
We can use all sorts of data. Lab experiments, animal studies, even cadavers. But the gold standard, especially for regulatory approval, is human data.
Right, because ultimately we want to know how these devices work in people.
Exactly.
Now, the source mentioned the prediction problem, which is about the limitations of using non-human data to predict human outcomes. Can you explain that a bit more for our listeners, especially those on the regulatory side?
Sure. Preclinical data is great for building initial confidence in a model, but humans are complex. We don't always react the same way as animals or cadavers. So while non-human validation is important, it's often not enough to prove a device will work safely and effectively in people.
So regulators really want to see that human data whenever possible.
They do.
Now, when we're comparing model predictions to real-world observations, there can be some differences, right?
Of course.
That brings us to uncertainty in validation. What are the main sources of uncertainty we need to be aware of?
Well, there's numerical uncertainty, which comes from the approximations in our calculations. There's input uncertainty, which means we can't know all the model parameters perfectly.
Things like material properties of tissues, right?
Exactly. Then there's experimental uncertainty, which comes from the limitations of our measurement tools, and the inherent variability in biological systems.
So no experiment is ever perfect.
Right. And finally, there's model form uncertainty, which acknowledges that our model is just a simplified representation of reality. There are always going to be some things it doesn't capture perfectly.
Makes sense. So uncertainty is everywhere.
Right.
And the source also talked about different types of uncertainty, aleatory and epistemic.
Right.
Why is understanding that difference important for someone in a regulatory role?
It helps us understand the nature of the uncertainty and how to deal with it. Aleatory uncertainty is like randomness or natural variation. We often model it with statistical distributions.
So things like the variation in blood pressure between people.
Exactly. Epistemic uncertainty, on the other hand, comes from a lack of knowledge. We just don't have enough data or understanding to be more precise.
Like not knowing exactly how a certain cell type will respond to a particular drug.
That's a good example. And epistemic uncertainty is very common in biomedical modeling because human biology is so complex.
So figuring out whether uncertainty is aleatory or epistemic helps us choose the right methods to quantify it.
Exactly.
Now, when we're dealing with those epistemic uncertainties, the source mentioned the area metric approach. How should someone evaluating an in-silico trial submission interpret the results of that analysis?
Well, the area metric is a way to measure how well the model's predictions match up with the experimental data. It takes into account the uncertainty in both.
And this is especially useful when we have those probability boxes because of epistemic uncertainty, right?
Exactly. A smaller area means the model and the data are in good agreement, so there's less model form uncertainty. A larger area means there's a bigger discrepancy, suggesting the model might not be as accurate.
So a well-defined area metric can be a good way to show that a model is valid, even when we're dealing with limited data.
Exactly. But it's important to remember that a small area metric doesn't always mean the model is perfect.
Right, especially if those probability boxes are really wide.
Exactly. That might just mean we don't know enough, not that the model is spot on.
So rigorous preclinical validation is crucial to narrow down those uncertainties.
Absolutely.
All right, we've talked about verification, validation, and uncertainty quantification separately. Now, how does it all come together in a real in silico clinical trial, especially in a way that would convince regulators?
The key is to have a clear framework, and the source outlined one that's based on a build, validate, predict approach.
Sounds logical.
It is, and it's all about demonstrating rigor and credibility.
Okay, let's walk through it. Step one is model build. It's not just about writing some code, right? What are the key things regulators would want to see in this phase?
Well, they'd want to see a detailed description of the model, the equations, the assumptions, the rationale behind it all. They'd also want to see how the model was calibrated, if necessary, using data that's separate from the validation data.
So keeping things separate to avoid bias.
Exactly. And once the model is built, it's frozen before the validation phase begins. No more tweaking allowed.
Makes sense. We need to be objective. All right, step two is preclinical VVUQ. What's the main goal here, and what kind of evidence do we usually gather?
The goal is to build some initial confidence in the model. We do that by testing it with non-human data, like benchtop experiments, animal studies, or cadavers.
Anything to get a sense of whether the model is on the right track before going to humans.
Exactly. And we start quantifying the uncertainties too, even though we might have limited data at this stage.
So it's about building that foundation of evidence.
Yeah.
Then comes clinical planning, where we design the in silico trial itself. What are the special things we need to include in the protocol from a regulatory standpoint?
Well, we need to be very specific about the model. We need to describe it in detail, define the quantity it's going to predict, and explain how that relates to the clinical endpoints that matter for patients and for regulatory decisions.
So we're linking the model's output to real-world clinical outcomes.
Exactly. We also need to decide how many patients we'll include in the first human cohort and what we'll do if the model doesn't validate well.
Like having a backup plan.
Exactly. And we need to define the criteria for model credibility up front, as well as the procedures for validation and how we'll use the model to make predictions if it passes validation.
So everything is transparent and predefined.
That's the goal.
All right, step 4 is perform first stage of the clinical trial. Why do we need this initial group of real patients in an in silico trial?
It serves two purposes. First, it gives us data on clinical endpoints that the model might not cover.
Things the model can't predict.
Right. And second, it provides the human data we need to finally validate the model.
So we're using this first group to check the model's work. And the source emphasized the importance of blinding in this phase. Why is that so critical?
Blinding is crucial to prevent bias. The people running the simulations shouldn't know the real patient outcomes, and vice versa. This way, no one can tweak the model or interpret the results to make them look better than they are.
It's all about objectivity.
Absolutely.
Step 5 is final model validation using human data. This is the moment of truth, right?
It is. We take the data from the first cohort and rigorously compare the model's predictions to the actual patient outcomes. And we refine our estimates of the model's uncertainties.
So we're seeing how well the model performed in the real world.
Exactly.
And then comes step 6, adequacy decision. Based on that validation, we decide whether the model is good enough. What goes into that decision from a regulatory standpoint?
It's all about comparing the validation results to the criteria we define in the protocol. If the model's performance meets those criteria, if the discrepancies are small enough and the uncertainties are within acceptable limits, then we can say the model is adequate.
Pass or fail.
Pretty much.
And if it passes, we move to step 7, model prediction. This is where the real power of in-silico trials comes in, right?
Exactly. If we have a validated model, we can use it to make predictions for a much larger virtual cohort of patients. And because we validated it with human data, we can be more confident in those predictions.
So we're using the model to explore a wider range of possibilities, maybe different patient groups or device variations.
Exactly. And we're carrying forward all the uncertainties we've quantified so we have a realistic picture of how reliable the model is.
But what if the model fails in step 6? That brings us to step 8, second human cohort. How should regulators view this backup plan?
It's all about managing risk. If the model doesn't work out, we go back to a more traditional approach. We enroll more patients and gather the data we need through a regular trial.
So it's a safety net.
That's a good way to put it.
Right. The source gave us a fictional example involving a Medtronic neuromodulation lead. Can we walk through that to see how these principles might apply in a real case?
Sure. Let's say Medtronic designs a new lead for sacral nerve stimulation, and they're worried about it migrating after implant.
So the lead moving around inside the body.
Right. And the clinical endpoint they're focused on is the complication rate related to that migration. They want to make sure it's below 5%.
Okay, so they want to keep the complication rate low. How does the model fit into all this?
They build a finite element model to simulate how the lead behaves in the body. The model can predict how far the lead tip might move.
But the model is predicting a distance, and the clinical endpoint is about complications. How do we connect those two?
That's a great question. In this example, they plan to figure out that relationship, how migration distance relates to complications, during the initial human cohort. They'll enroll 150 patients and track both lead migration and complications.
So they're using that first group to ground the model in real clinical data.
Exactly.
Now, before they even get to those 150 patients, they do some preclinical validation with cadavers. What's the point of that?
It's about building some initial confidence in the model. They compare the model's predictions to how the lead actually moves in cadavers.
But cadavers aren't exactly like living people.
Right, there are limitations. The sample size is small, and the tissue properties aren't the same as in a living person, but it's still a valuable step.
So it's about getting some early validation, even if it's not perfect.
Exactly. And they use those probability boxes to account for the uncertainties in tissue properties. They compare those to the experimental data from the cadavers using the area metric.
And how did it look?
The area metric was small, which is encouraging, but it wasn't enough to justify skipping the human trial altogether.
So they moved on to the In-Silico trial with those 150 patients. What happened there?
They found 5 potential complications in that group, and by analyzing the imaging data, they could start to see a link between how far the lid moved and whether a complication occurred.
So they're starting to define that relationship between migration distance and complication rate.
Exactly. And they set a threshold for migration distance, like 8 millimeters, beyond which the risk of complications seemed to go up significantly.
Okay, so they're setting a clear limit based on the clinical data.
Right. And at the same time, the simulation team is running the model for those 150 patients, blinded to the real outcomes.
And then comes the final validation, where they compare the model's predictions to the real patient data.
Exactly. They use the area metric again and find that it's well below the predefined acceptance criterion.
So the model passed the test.
It did.
And that means they might not need a much larger traditional trial for this specific aspect of lead migration.
That's the hope. They can move on to making predictions for a larger virtual cohort using the validated model, and they carry forward all the uncertainties so they know how much to trust those predictions.
So they're being transparent about the limitations of the model.
Exactly.
All right, let's talk about some of the broader implications of all this. The source mentioned the FDA's least burdensome principle. How does that relate to in-silico trials?
Well, if a validated model can provide strong evidence of safety and effectiveness, it could be a much easier and cheaper path to regulatory approval than a big traditional trial.
So it could streamline the process.
Exactly.
But the source also pointed out that the framework we discussed is pretty strict. If the model fails, it's basically out.
That's right. It's all or nothing.
Which seems risky if you've invested a lot in developing the model.
It is. And there are other approaches, like Bayesian methods, that allow for more flexibility and model refinement.
Interesting. So there are different ways to do this. What about the challenge of getting people who aren't engineers or modelers to accept these methods?
That's a big one. Not everyone is familiar with the VVUQ or comfortable with computer-generated evidence. It takes time to build trust and educate people.
And we talked about the ethical considerations of getting human data for validation, like needing extra imaging in the example.
Right. We always have to weigh the potential benefits of the model against any risks to the patients involved.
And that link between the model's output and the clinical outcome, the transfer function, is important too, right?
Absolutely. Ideally, we want to validate that link separately using existing data or preclinical studies to make the whole trial more credible.
And finally, we talked about the area metric and how it can be misleading if there's a lot of epistemic uncertainty. Can you remind our listeners about that?
Sure. A small area metric doesn't automatically mean the model is great. If the probability boxes are very wide because of a lack of knowledge, the model might just be getting lucky.
So preclinical validation is crucial to reduce that uncertainty as much as possible.
Exactly.
All right, let's wrap this up. What are the key takeaways for our listeners who are dealing with the regulatory side of medical devices and thinking about in-silico trials?
The bottom line is that in-silico trials have huge potential, but they need to be done rigorously. We need strong VVUQ to show that these models are credible and that their predictions are reliable, and we need to be transparent about the uncertainties involved.
So it all comes down to trust, to showing that these models can truly reflect reality. Here's a final thought for our listeners to ponder. As biological systems get more complex and our models get more sophisticated, how do we balance the power of in-silico trials with the need to protect patients and ensure the integrity of medical device evaluation?
That's the million-dollar question.
It is. Thanks for joining us on the Deep Dive.
My pleasure.